repression. Glucose is a catabolite, lactose is a catabolite. Anything that is metabolized, especially in catabolic reactions, is called a catabolite. So it's just a fancy name for a biomolecule. But the phenomenon of catabolite repression is when one nutrient, aka glucose, is repressed by the presence of a more favorable nutrient. Sorry. The more favorable nutrient is glucose repressing the lac operon. So glucose is preferred, and so it is used first. A biphasic curve of the culture on two carbon sources is often called dioxic growth. Dioxic means that there are two distinct stages of growth. So this is figure 10 in your text. And let's see. Yeah, I'm going to talk about that in just a second. But in this first scenario, understand that both glucose and lactose are present at time 0. So right at this point of the experiment, you expose cells to both glucose and lactose. And then you track cell growth by how dense the culture is. And you also track the concentration of lactose and glucose. And so this first phase of cell growth is growing at the expense of glucose. So glucose is utilized first, and the lactose concentration remains the same. And lactose only is started to be used after this little lag phase. What do you think is going on during that lag phase? The synthesis of those lac genes. And once those genes are expressed and those enzymes are present, now glucose is exhausted. There's no more glucose. Now the cell starts using lactose. Okay? And so then it has a second phase of growth. That's why it's called dioxic. So my little point here is that they really should show the shape of that curve, the slope of that line, to be less when the cells are growing on lactose than when they are growing on glucose. Glucose is a better carbon source. They grow faster on glucose than when they grow on lactose. And so adding that little dashed line just kind of emphasizes why does it prefer glucose? Well, it can grow faster using glucose than when it has to switch to growing on lactose. And so I just add that. Any kind of a different shape, just a lower slope of that line will represent growth on lactose. Okay, so that's what's going on in A. A different experiment is at the beginning. Now we're looking at not just overall cell growth, we're looking at the activity of that first enzyme in the lac operon, beta-galactosidase. And you have at the beginning of this experiment just lactose. Okay? So cells are growing, and while they are growing, they are producing more and more beta-galactosidase enzymes. Now in the experiment, you still have lactose, but you add glucose. And that is then going to shut down gene expression of the lac operon. And so beta-galactosidase represents the synthesis of the lac operon. Glucose is added, and it stops the synthesis of those lac genes. Whereas in, say, control cultures, you don't add glucose, beta-galactosidase continues to be produced. So these are some of the ways that this whole pathway was figured out. Kind of experiments with cells and looking at enzyme assays. Okay, any questions on these plots and what kind of sense they make? Okay. Let me just introduce you to text that explains this figure. Okay, so we'll just cover the text today. And you say, gosh, I don't understand this text. Well, it really is exemplified in kind of a nice explanation just right there in the figure. So as I've said many times, when a number of operands are turned on coordinatedly, in a coordinate fashion, they're called a regulon. And these operands are scattered around the chromosome with related functions. So they're coordinately controlled by a single repressor. So we saw that in the SOS response. The LEX-A regulatory protein turns on or off 5 or 6 different operands. So we've been talking about regulons, and we've also been talking about sigma factors. So we have regulatory proteins that bind to the operator, but we also have sigma factors that bind to that RNA polymerase and lead it to its site where it is going to express those genes. So that is another layer or another type of regulation. So we're talking now about, again, we've talked about sigma factors. We're bringing them back up in the context of regulating gene expression. And so cells can regulate the synthesis or activity of the sigma factor that then directs the expression of all those genes. And so sigma factors are also regulatory. So we're going to be talking about the heat shock response in E. coli and really an awful lot of bacteria. We have a heat shock response that is upregulated with stress. And so I'm just going to describe this, and we'll look at this on Monday and look at this over the weekend. Under low heat conditions, there's a secondary structure that forms in the messenger RNA that does not allow the ribosome to attach. So we are now looking at a combination of transcriptional control, and right now we're talking about translational control, because if that ribosome is already on the messenger RNA, we're talking about translation. But it cannot, because of this secondary structure that stops the ribosome from binding. So that's under low heat conditions. Under high heat conditions, that secondary structure is actually melted. They become dissociated, and that particular that particular sigma factor is now translated. So you make the sigma factor, you make the protein. And then the second factor is this, the fact that heat draws these chaperone proteins away to repair those proteins. So this phenomenon, we're going to talk about chaperone proteins and sigma factors in this particular figure 17. So look at this figure 17 over the weekend, because it brings a lot of things together that we've been talking about. Like I said, sigma factors, secondary structure, chaperone proteins, and now the context of stress response.